Hi class, in this video we'll talk about how to go from DNA to making your proteins and the words transcription and translation. I read all of your cards I received from you all from the last time we were in class together, and I'll try my best to cover all the questions I received in those cards. So one main thing is, what is DNA transcription and translation, and how can we relate to something that we know something about? Overall, the main theme of chapter 10 is how do we go from our DNA and how do we actually make our traits. We understand that from previous chapters that we get our DNA from our parents, and based on that DNA, we form our traits. And the traits that we make are based on the proteins we make. So the proteins we make dictates the traits we have, such as brown eyes or black hair. And we know that we don't just go from DNA to proteins. There's actually a middle step, and that middle step is making the RNA. And the type of RNA that we're actually making is called messenger RNA, or mRNA. mRNA, or the messenger RNA. And so overall, we have two major steps going from DNA to mRNA, and then from mRNA to actual proteins. And we know that going from DNA to mRNA is transcription. And we know going from mRNA to proteins is translation. And so I'm going to use an analogy to help us understand this process. And for example, our DNA contains all of our genes that we got from our parents. And so who we are is based on the DNA that we have. So in our analogy, we're going to take DNA and we're going to imagine we're at our kitchen and we want to make a recipe. And we want to cook a recipe. And typically, where do we go? We either go on the internet, or some of us may even have some recipe books. And so in a recipe book, there are many, many recipes that you can choose from. And so we're going to call DNA our recipe book. And in that recipe book are many recipes, from appetizers to salads to main dishes to desserts. Everything you want to make a meal is in our recipe book. And DNA is our recipe book because in our DNA it has all the genes that make us who we are. But when we want to cook something, do we want to cook everything in the recipe book? Probably not. We only want to make a few recipes at a time, and sometimes we want to make one dish at a time. So if you take the entire recipe book, you only want to make one dish at a time. So sometimes what people do is that they take the recipe book and then they write down the recipe on a card, so that they don't have to worry about the whole book being on the counter. Sometimes they actually take the recipe book and they write the recipe down on a card, and that card is what they lay on the table while they're cooking. Or they may even write it on a chalkboard. If they have the special chalkboard paint in their kitchen, they'll take the recipe and write it on their chalkboard. So basically, the idea of taking a recipe book but only writing down one recipe that we're actually going to use at the given time is the same thing as transcription, because in transcription, this is where we want to actually make a protein, but we start from our recipe book, which is DNA, but we only want to make one protein at a time. But before we get to that protein, we want to copy one recipe from that manual. And so when we do transcription is that process from going from your DNA molecule and making messenger RNA. This messenger RNA will later on be used to make a protein, but the messenger RNA contains information from one gene. So when we do transcription, we're taking information from one gene and we're making the messenger RNA from it. So again, this is again taking your DNA, but we only want to make a protein from your DNA, not everything in your DNA. So what we want to do is look at one gene in your DNA, and we're going to basically copy that information down. And so that is transcription. And when we do transcription, we make messenger RNA. And that messenger RNA is going to have the message or the code for actually building the protein that you want. So again, this is our major theme. We have our DNA, and eventually we want to make our trait, which is a protein. But the middle step is making messenger RNA. And so that's transcription. That, of course, in a eukaryote takes place in the nucleus. Don't forget that. And again, we want to make a protein, but we just want to copy the information down first separately. We don't want everything in the DNA. That's too much to do. To simplify it, let's just do one gene at a time. And so taking the information from one gene at a time and copying the information down in the form of RNA is called transcription. Now we're ready for the next step. So again, if we're following our analogy of cooking, we're in our kitchen, we had a recipe book, we took one recipe, wrote it down on a piece of paper, that was transcription. And at that point, I'm going to read the recipe, grab the materials I need, and actually make that dish. So going from the actual recipe to actually making the dish is translation. So if the mRNA is basically the recipe on a card, then in translation, this is where I actually make the dish. And the dish is based on the instructions that were contained in the recipe written down on a card that came from the recipe book. So translation is the process of going from messenger RNA to making the actual protein, and the instructions on making the protein, they were contained on the messenger RNA. And of course, the message or the code came from the entire recipe book, which is basically our DNA. And so this process in the eukaryote takes place out in the cytoplasm. The main idea is that translation takes place outside of the nucleus. And you can say, for example, that of course the actual organelle that actually makes the proteins based on the messenger RNA is the ribosome. So of course we know that to be true. But where are these ribosomes? Are ribosomes in the nucleus? No. Ribosomes are out in the cytoplasm. You could even say the rough ER, which is fine. But again, the rough ER, is that in the nucleus? No, the rough ER is out in the cytoplasm. So generally we say that translation takes place out in the cytoplasm. This is where we take the message that's contained in the messenger RNA. This message tells us how to actually make the protein, which will basically give us things like our eye color or hair color. And so in our analogy, translation again is now preparing the actual dish, putting the dish together based on the information written down on our recipe card, which was our messenger RNA, and that messenger RNA came from the entire recipe book, which was DNA. So let's go through this exercise where we're going to practice doing transcription and translation. And you'll be given a DNA molecule like this one, and it's going from 5' to 3' direction, and we're asked to do transcription. So what is the mRNA that is transcribed? And the rules are similar to DNA-based pairing, but again in RNA we know that there are no T's in RNA. We have uracil instead. So let's see how this works. Of course, it's still going to be anti-parallel. This goes back to the first video we had from chapter 10. So if this ends starting with 5', then this strand here will have to be 3'. And you can just start base pairing as you normally do. So If you see a T, an A will go there, and A will go there. Now, normally, if you have an A in DNA, we would put a T if we're doing DNA, but again, we're doing transcription, which means we're making RNA. So if you see an A in DNA, of course, we need a U in its spot. So advancing on, need a G there, a C, a G there. We have some AAA, so of course, this is going to be UUU. Then an A there. We have an A here, we need a U there, and an A, and then 5 prime. So that was easy. I think so. So we did transcription. But remember back in our previous video, when we make DNA from the 5 prime to 3 prime direction. Well, this is the actual same direction where we make RNA. We make RNA also in the 5 prime to 3 prime direction, and it's okay to write it out this way because it's the easiest thing to do. But all you have to do is just write your RNA starting with the 5 prime end first and ending with the 3 prime. Again, 5 prime means where I start my sentence, and 3 prime is where I have my punctuation. So my answer for this part will be 5 prime and AUG. And where did I get that from? 5 prime AUG. Next will be GCC. GCC. Next would be GAU. So GAU. Next would be UUG. Next would be CGA. And last would be UAA. And then end with your 3 prime. So that's your messenger RNA from your DNA molecule. And the next step is asking you to now do translation, make the actual protein. And we need to use our codon chart. So let's go over some things. Codons, what are codons? Where are they found? How do they play a role in all of this? Well, basically, when we make messenger RNA, every 3 bases in our code, they are called codons. And codons, they actually code for amino acids. So again, if we're trying to make proteins, the building block of proteins, they are amino acids. And within this code, in the messenger RNA, every 3 bases codes for an amino acid. So we call these 3 bases, we call them codons. And codons code for amino acids. And we have a codon chart. You will be given a chart for your exam, so you don't have to memorize the chart, but just know how to use the chart. And as you see, every 3 bases codes for 1 amino acid. And so we're going to start translation in the 5 prime to 3 prime direction, and it's already in 5 prime to 3 prime direction, so we're ready to go. So first, let's look up the first 3-letter code, AUG. So the first letter is an A, second letter is a U, and so basically I'm going to connect where my fingers would meet. So I'm going to end up somewhere in this area here, and AUG stands for methionine. So we can write MET. And typically all proteins start with methionine. This is their start. The next is G, that's a C, GCC. So first letter is a G. Next letter is a C. So basically I'm going to end up somewhere in this box here, and GCC is ALA. Okay, let's go on to GAU. So first letter is a G, second letter is an A, so I'm going to end up in this box here, and GAU stands for ASP. Now, U-U-G. First letter is a U, second is a U. So I'm going to end up in this box here. U-U-G is L-E-U. Next codon is C-G-A. First letter is a C. Second letter is a G. Next one is an A. So C-G-A is A-R-G. And the last one is a U-A-A. First letter is a U. Second letter is an A. So I'm going to be in this box here. U-A-A is a stop. Stop codons, there are 3 stop codons. You don't have to know what they are because you'll be given a chart, but there are 3 stop codons. They just tell us to stop translation. And so this is the actual protein that you just made. So I hope this video was helpful in going over transcription and translation, the analogies. I hope we're helpful. The actual process of how to do it, I hope we're helpful as well. And thanks for watching.